Hi everyone! In this lecture we're going to be talking all about different types of genetic technologies. This lecture is sort of covering chapters 19 and 20. I would say most of this content is coming from chapter 19 in your textbook, and just a little bit is going to be coming from chapter 20. So on the syllabus, you'll see that this is chapters 19 and 20, but again, most of this is coming actually from chapter 19. And also, I should warn you that a fair bit of this information is information that I have added. It's sort of extraneous to your textbook, which just means that it's not necessarily found in your textbook, but it's found here in this lecture. So if you see a particular point in here and you say, hey, I don't see that in my book, it's because I've added some more information, specifically when we get to the section on genetically modified organisms. All right, so I'm getting ahead of myself. What are we going to cover in this lecture? We're going to talk about a lot of applicability of the things that we have learned up until this point. So, knowing what we know about DNA and genes and traits, now we can talk about all of the really great applications of this as far as the technologies that we are utilizing. So we're going to talk about genetic engineering. We're going to talk about modified organisms, cloning, gene therapy, and gene patenting. So lots of different topics here and things that hopefully you have come across in maybe some other aspects of your personal lives. Or if you haven't before, at least now you'll start to sort of recognize these things that we're going to discuss. So first of all, let's talk about this concept of genetic engineering. So what does it mean to genetically engineer something? Well, if you think about engineering in a traditional way, so we engineer a bridge, we can build a bridge, or we need engineers to help us build skyscrapers and structures and things. So we have to manipulate materials and we have to sort of put them together to build things. Genetic engineering is kind of the same concept, except we're manipulating an organism's DNA. There can be a variety of different things that we can do to manipulate DNA. We can combine DNA from different organisms. We can do what's called gene splicing, which is basically just inserting genes into other places. And we can also do direct genetic modification. So we can change bases and letters. We can go from A's to T's and C's to G's or whatever we need to do to actually modify things. So why would we want to do this? Why would we even want to go down this path? Well, there's a whole field known as biotechnology that's very specific about how we are using cells or biological molecules for specific applications in the real world. So the biotech industry is one that it's new, but it's not really new. Biotech companies have been around since the late 1970s, but it's really over the past 15 to 20 years that we have seen a boom in this type of sector, if you will. And there's all kinds of things and new and exciting things we are learning every day about what we can do when we alter or modify cells or molecules. A lot of times biotechnology is working with something called transgenic organisms. And a transgenic organism is anything that has DNA from a different species. So if we take DNA from a fish and we put it into a tomato plant, that's a transgenic organism we've just created. If we take bacterial DNA and we put it into a corn plant, that's a transgenic organism because it has DNA from different species. We can also look at something called just the recombinant DNA itself. So the DNA that we're talking about in creating these transgenic organisms, it starts with recombinant DNA, and recombinant DNA is when you combine DNA from multiple organisms. So to create a transgenic organism, you need recombinant DNA. And again, the reason we can do all of these things, the reasons we can take snippets of genetic information from one species and one organism and put them into another is because of the universality of the genetic code, right? So all of our cellular equipment, it doesn't matter what you are, even bacterial cells have ribosomes. they can make proteins, they all read the code the same way. And so those codons stand for the same amino acids in fish and frogs and humans and plants and bacteria, and it doesn't matter what it is. We're all using the same code. So we can create all kinds of different things. So in this case, they took the gene for green fluorescent protein. It's called GFP, is how we shorthand that. And we took that gene, it's from a jellyfish, and we put it into mice. And we actually were able to create mice that sort of glowed with this green fluorescent protein. And you might say, well, why would you want to make mice glow? The idea here is that in order to test the technology to make sure that it worked, we needed to have a very visible product, something that we could immediately tell whether it was working or not. And this green fluorescent protein was an immediate indicator of the success of creating transgenic organisms. All right, so let's talk through the steps very briefly about how we make recombinant DNA molecules. And there's several different steps here, and give me just a moment because I might go to a picture first. Okay. Here we go. I'm going to go to this picture and then I'll work back to the slide that I just skipped. So here's what we do. We want to take a specific segment of DNA. Let's say it's that green fluorescent protein gene that we just talked about from the jellyfish. Okay, so here it is. Okay, so we got some donor DNA from a jellyfish. All right. In order to use it, we need to cut it apart into small pieces. All right, so we're going to use something called a restriction enzyme. Restriction enzyme is a special enzyme that's going to cut our gene up into fragments, and we can design these enzymes to cut the genes exactly where we want it. Okay, so we can cut it in very specific places and just cut out that jellyfish gene for green fluorescent protein. Okay, we're going to combine it with something. And what we're going to combine it with is called a vector. A vector is any small piece of DNA that we can use like a shuttle to carry our gene into an organism of choice. Okay, so we can't just put the DNA in there by itself. We need like a shuttle, like a taxi to carry it for us. So viruses and bacteria are great at doing this. They're great at shuttling our genes. So we're going to take what's called a plasmid, which just is a circular piece of bacterial DNA, and we're going to use the exact same restriction enzyme that we used on the jellyfish DNA, and we're going to use it on our bacterial DNA. And what this does is it creates these, we're going to call them sticky ends. It simply means that we have these ends that are going to be compatible with each other. They're going to stick to each other. So when we combine our jellyfish DNA and our vector DNA, our plasmid DNA, it's going to create what are called recombinant DNA molecules. Okay, and again, why do we call it recombinant? Because we have just combined DNA from two entirely separate species, a jellyfish and a bacterium. And we've created this nice little shuttle for our gene. And then we're going to get that gene into our cells of interest. Okay, so backing up just for a moment, this page here is just, again, going through exactly the same steps that I went through, but it's kind of helpful to see a picture of it first. So again, this information is the same. We cut it with restriction enzymes, we cut the vector with restriction enzymes, and then those ends are going to stick together because they're complementary. All right, so now we have these recombinant DNA molecules. Now we need to get them into our species of choice. So in this case, we want to get them into those mice. Okay, so how are we going to do this? So how are we going to stick, and it would be mice embryos, by the way, that would be doing this with. How are we going to put this DNA in there? Well, there's a couple of different ways to do it. You can do something called electroporation. And electroporation is where you use electricity to shock the embryo to create like a little tiny hole in it, and then you flood it with your recombinant DNA. You can actually use teeny tiny little needles. It's called microinjections. You can use something called a gene gun, which is particle bombardment. There's these little bullets. They're actually little pieces of gold that you coat with your DNA, and then you shoot them into the embryo. It's called a gene gun. Or then you can use what's probably the most common method. You use a virus. And viruses are great because viruses infect cells for a living. This is what they do. This is their job. So why not use what we already have to get the DNA into our cell to begin with? All right. So that's the process of creating these transgenic organisms. But again, why would we do this anyway? Well, there's a whole host of reasons why we manipulate genes in the biotech sector. Most of what we are doing right now is to create proteins of value. So there are a whole host of what we call biologics that are on the market right now. Biologics are medicines that are from human proteins. So we're actually using human proteins to treat things. So if you know anyone who's diabetic and they take insulin, the insulin they're taking is human insulin. They're getting that from bacteria cells. So basically, we have created these large, giant vats that have bacteria in them, and we've inserted the human gene for insulin. We can also make human growth hormone. We can use this for vaccines, beta blockers, statins. You name the biologic, the living molecule, the protein, if you will, that can be used for therapeutic and medicinal purposes, and we can engineer a bacteria to make it for us. We also do it for experiments in science. So the more we learn about our genes, the more we want to find out what happens if we stop producing a protein. What happens if we produce too much of a protein? What are the consequences of them? So there's definitely a lot of scientific research in this field. So these are all the drugs that can be produced using recombinant DNA technology. And again, if you sort of look at the different things here, and again, these are all made from bacteria. So it's really amazing what we are able to accomplish now with this recombinant DNA technology. So we can also do something a little bit differently. Instead of using bacteria to create the proteins of interest, we could actually do something with animals as well. So an even more efficient way to express some recombinant genes is actually in the body fluid of a transgenic animal. So you need big vats of bacteria and lots of bacteria in order to produce very small quantities of these proteins. But what if you could get that same protein from the milk of an animal, for example? So they have been using, and it's mostly in experimental phases right now, sheep, cows, and goats. They have been putting human genes into them, and these animals will produce the gene products in their milk. So we can do this with clotting factors, with something called clot busters and collagen. So it works for a certain subset of proteins and not necessarily all proteins, but it would be a much more efficient way to get these human proteins versus using sort of the traditional bacterial method. We can use it for antibodies, a whole host of things. So transgenic animals are also great models for human diseases. So we can actually put in a mutant form of a gene, and we can see what drugs might be able to work on the animals first before we test them in humans, for example. So beyond the medical community, the other place where you're really going to see this type of technology, this biotechnology, if you will, medicine and agriculture are sort of the two big ones. And agriculture, we talk mostly about this concept of genetically modified organisms. So even the transgenic organisms that we were talking about, that's a GMO. GMOs just are most often used to represent plant crops. That's probably what you've heard them for. So a genetically modified organism is, again, any organism that contains recombinant DNA. And we have been doing this for a whole host of plants for the last 25 plus years. So we can engineer insect resistance into our crops. We can make them herbicide resistant. We can make them virus resistant. So there's a whole host of reasons why we put DNA, foreign DNA, if you will, foreign just meaning different DNA, into our plant crops. And these are all things that we have worked on. So again, delayed ripening, altering the composition of things. All of these are crops that we have engineered for very specific purposes. So if you're not aware, here in the United States, at least 70% of processed foods are going to contain at least one genetically modified ingredient. So what that means is if you go to the supermarket and you go to the area of the supermarket that has anything in a box or a bag, so anything that's packaged, something that's been processed, there's a 70% chance that one ingredient in that ingredient list is going to contain a genetically modified ingredient. We don't separate genetically modified varieties of our crops from the non-genetically varieties of our crops. So they're sort of all thrown together, and the processing plants will get corn or soy or whatever it is that they need, and they don't separate whether they're genetically modified or not. So the farmers mix them. If it's mixed up in harvesting, shipping, storage, it doesn't matter. Okay. So the three major crops that we engineer here in the United States are soy, cotton, and corn. So those are the three major ones. Soybeans, and again, my statistics are a little out of date, so forgive me. So this was as of 2013. 93% of soybean crops are genetically engineered, and they're being engineered for herbicide resistance. So you might think, who cares about soybeans? I don't eat soy. I'm not a tofu eater. It doesn't matter if you eat tofu or not. Soy is found in all kinds of things, mostly in the form of soy protein, soy isolate, lecithin, and isoflavones. So if you turn over a package of anything, you're probably going to find that ingredient in there. Corn is the next one. Again, 90% of the corn crop is genetically engineered, mostly for herbicide and insect resistant. And again, you name it, corn flour, oil, corn syrup, starch, gluten, the list goes on and on and on and on. Corn and soy is in just about everything we eat. Cotton is the other one. Again, 90% of the crop that we're growing. Other things we engineer, some types of vegetables. They've been approved by the FDA, but you're really going to find them in very limited quantities here in the United States. Again, it's mostly the soy and the corn, and then we don't eat cotton, of course, but cotton is the other big one. So this kind of brings up an interesting idea about product labeling. Here in the United States, the FDA regards DNA and proteins added to conventional foods by modification as being safe. So because they're generally regarded as safe, there is no pre-market safety review. So we don't test these things on other animals first to see whether or not we should be eating them. We just figure, it's DNA, it's protein, we're probably okay. So the FDA would only make labeling required if there was a known consequence. So it was less nutritious, or it was more toxic, or there was more risk of having an allergic reaction. And so because we don't have any concrete evidence of those things, they don't require any labeling. So in one of the things you're going to explore in the activities of this week, there's lots of pros and cons when it comes to genetically modified foods. So we get more crop yields, and they are better able to tolerate stress. They improve pest resistance, and we get new growing techniques. But there's also a lot we don't know. We don't know about the health risks long term of consuming these. We don't know what the consequences are if these genes get out into nature versus staying in our crops. So you'll explore this topic more in the activity this week. Last thing I want to talk about quickly is cloning. And there's more here, but I'm just going to kind of talk about cloning before we wrap things up. So cloning is when you produce a genetically identical DNA sequence, a cell, or an organism. And you can make clones in one of two ways. You can take embryonic cells and separate them to produce two distinct offspring instead of one. Or you can use something called somatic cell nuclear transfer, and this is when you are replacing an egg nucleus with the somatic cell of another organism. So let's talk about somatic cell nuclear transfer and how we create a cloned pig. Okay, so let's say our pig here on top, piggy number 1, is the adult pig we want to clone. Okay, so we're going to take the skin scales from this adult pig, all right, and then here's our adult female. Okay, our adult female is going to be basically she's going to give us one of her egg cells. So we're going to take an unfertilized egg from her, and we're going to remove the nucleus from it. We're going to take the nucleus out of it, so there's no genetic information in here. and we're going to take the nucleus from the skin cell, from piggy number 1, and we're going to fuse these two together. Okay, so we get one cell. It's an egg cell from our female, but it has the DNA from piggy number 1 up here. And then what we're going to do is we're going to basically get the cell to divide, again, using sort of the same electricity, into an early stage embryo. And then we're going to put it into piggy number 3. And piggy number 3 is just she's a surrogate mother. That's all she is. And she's eventually going to give birth to our cloned animal. So our cloned animal is a genetic copy of piggy number 1. So again, that's called somatic cell nuclear transfer. We're taking the nucleus from a somatic cell, a skin cell, putting it into an empty egg cell, and then implanting that into our surrogate. So why would you do this? Well, as it turns out in nature, we do this a lot. Plants in particular are really great at making copies of themselves. They do it all the time. A lot of our produce that we use are clones, grapes, bananas, potatoes, you name it. Female insects can sometimes do this too. If there aren't enough males around, they'll just make copies of themselves, basically. We call it parthenogenesis. And then, again, in humans, we can do this, we could clone agricultural animals if we wanted to. There's been some discussion of cloning extinct or endangered species, but again, the costs and the benefits of that are somewhat debatable, to say the least. There's even some companies that will clone your pet if you want them to. If you want to go and look up that information, you're more than welcome to, but there are companies that will offer you the ability to use the DNA from a dearly beloved pet who might have died, and they can clone it for you. Not here in the United States. The companies aren't located here, but there are companies that can do that for you. I'm going to talk really, really briefly about gene therapy here in the last couple minutes, because I don't want to keep this going too much longer. But gene therapy is the idea that we can replace non-functioning genes that are causing a genetic disease with a normal gene. That's kind of the promise of it. And the idea is that if you had a disease caused by a single gene mutation, we could actually replace that bad gene with a good gene that works well, and we could treat your disease. That's kind of the idea behind gene therapy. So it was first used in 1990 to treat something called SCID, which is an immunodeficiency disorder. It's only approved for use in somatic cells. That means we can only treat an individual. We can't treat their offspring potentially in the future. So here's kind of how it works. You would put the gene into a virus, and then you would have the virus infect the cells of the patient, and they would put in the new gene that's functioning, and you'd make the protein, and hopefully you would be okay. It doesn't work very long is the problem with it. There have been some very strong immune responses because you're using viruses, so it doesn't really work all that great. And again, there's been some problems with some of the patients. And again, you can only use this for a single gene disorder. You can't use it for anything that's caused by more than one gene. So there's clinical trials being conducted all over the world looking at how we could treat things, blindness, cancer, melanoma, different types of leukemia. So there's a lot of promise for gene therapy, but there hasn't been a lot that's been delivered. So it's more of an idea that potentially could be very powerful in the future. Last but not least, gene patenting. You can put a patent on a genetic sequence, its usage, or its chemical composition. There's been about 3 million genome-related patents filed. You can patent fragments, you can patent stem cells, you can patent proteins, as long as it's new, useful, and not obvious. So you can't just patent any old DNA sequence. You have to use it for a diagnostic test or a drug or something new and improved. So I'm kind of going through this really fast, but don't necessarily worry about it too much. Here's the technology timeline for as far as gene patenting goes, and we're done.